What year it is since SIMI started in 1982.
Oh, 42nd year.
So 42nd year, but the 37th year for the summer program for high school students and later undergraduates. And this has been funded by the National Institutes of Health and several foundations. And the institutes at the National Institutes of Health, this could be in the recording, are the Heart, Lung, and Blood Institute, the Neurological Diseases and Stroke Institute, and the Childhood and Development Institute. So you had different sources of funding depending on the type of research that you're doing in the labs that you're working in. So we're going to get started with the final presentations, and the videos will be shown today and tomorrow.
Okay, thank you.
Personal privilege, Dr. Matsunaga, who's been a recipient of the Kettle Award for mentoring and has been a mentor really since the program began.
Well, maybe not quite that long, but for a while. But I just wanted to give you an idea. I mean, this is, I guess it's the 43rd year or something like that. You might have to go in on the other side here. This has a little-
Go ahead up.
Okay.
There, if you wish. This is an impromptu comment.
Yes. To be funded every year for 43 years is just a phenomenal thing. You know, I mean, I've been funded, I've been here for 17 years, and I was in the private sector for 16 years. And I was funded on and off, you know, for those 30 years, but never consecutively. And to be able to be funded like that, I wish I had a program like this when I was a kid. I would have kind of been motivated a lot faster than I actually was. But I think this is just a fantastic program, and I think you have a fantastic mentor who, yeah, Dr. Wood here.
I'm mentor-in-chief.
Yeah, she's the boss, yes.
And incidentally, yesterday we got word that we got an outstanding score on our medical student program. So it's likely that we're going to go to 46 years with funding for the MSRP. But we don't have the money in hand, but the score was outstanding.
And you should really, really be happy and proud that you got into the program like this, because it is incredibly difficult to be able to maintain something like this for all these years. So my hat's off to you, Marlise. This is just really something, you know.
Okay.
I wish I could last.
Well,
half
virtue
of
is its own reward.
I want to give her a hand right now.
All right, our first student for the 2024 SIMI High School undergrad student final presentations today will be Kamal Rizal, and he is representing Flowing Wells High School here in town, and he was mentored by Dr. Terry Matsunaga. And Kamal, go for it.
My name is Kamal Mode, and this is the matter of minuscule things. So my central question during my time here was, what is a cheaper way to obtain high quality inner bodily and cellular images? So instead of using expensive methods like MRI or CAT scans, we wanted a different way to get those images. So our potential answer that I was shown by Dr. Terry Matsunaga was ultrasound through perfluorocarbon microbubbles. So one of the main lessons I was taught by Terry Matsunaga was to understand a concept, it must be broken down to its simplest parts. So in this presentation, I'm going to break down the big questions into smaller questions in order for it to be more understandable. So how do we use ultrasound with fluorocarbon microbubbles? Well, the microbubbles are able to reflect acoustic energy generated by the ultrasound machine, and then we're able to get the images produced. Now, these microbubbles can only fit in the human vasculature. So when we're trying to get inside the human tissue, it has to be like 1/6 of the size of the microbubble. So we have to condense them into nano droplets and expand them within the flesh in order to get an image of it. So, in order to create the microbubble formulation, we have to first weigh the solutes. We have to mix the solutes and the solvents, and then we have to purge it in order to inject the perfluorocarbon of our choice. So, what are we mixing? Well, the solutes are the lipids DBPE, DBPC, and DPPA, and our solvents are propylene glycol, saline, and glycerol. Now, the order in which we mix these solvents are incredibly important, since if we started dissolving the lipids in low solubility solvents, they would clump up and turn into large particles. So we have to go in order slowly from high solubility to low solubility in order to efficiently dissolve the lipids within. So why do we use perfluorocarbons? Well, they are non-reactive to human tissue, which means they're safe to use in a human environment. And octafluoropropane and decafluorobutane are the most commonly used perfluorocarbons because they are able to stay in their gaseous forms while under body temperature. So how do we generate the microbubbles and turn them into nanodroplets? Well, first we have to shake them up and get them into a foamy white substance. Then we have to cool them, and then we have to pressurize them to get them back into nanodroplets. So how do we know they are the right size? Well, after shaking them, we again get this foamy white liquid, which is actually just made of very small microbubbles, since the human eye can only see down to 15 nanometers or 15 micrometers. And so when we can't see the physical actual bubble, it has to be less than 15 micrometers, which is our goal. Now, why do we decrease the temperature of the bubbles, and why do we pressurize the bubbles? Well, through the law of ideal gas, temperature is relative to volume, and pressure is inversely relative to volume. So when we decrease the temperature and increase the pressure, we are able to condense the microbubbles into nanodroplets and turn them back into liquids. So how do we categorize the perfluorocarbon nanodroplets by size and charge? Well, through the zeta sizer, we are able to rapidly measure the size of the nanodroplets and find the zeta potential. Now, what is zeta potential? Well, zeta potential is the charge on the surface of the microbubble. Now, this is important so that we are able to attach various compounds onto it that are able to latch on to other cells and organs in order for it to deliver to that place. Now, we always want to improve this model because it's far from perfect. So one of our compounds that we use to coat the microbubble shell was PLGA, which is a synthetic polymer that is able to basically weave around the bubble and create a hard shell around it. We also tried hyaluronic acid, which is a hydrogel that weaves around the microbubble with a soft shell. Now, my ongoing questions from my time here was, what other compounds can we apply to the microbubble in order to reinforce it? Another question was, what happens if we combine multiple compounds together in order to make like a hybrid shell? And my last question is, what other alternatives can we use for ultrasound other than the microbubbles? And that's it.
One question from the audience? Get your question skills going. Terry, you have 30 seconds if you want to make a comment, if not.
Oh, okay.
30 seconds.
That's very good.
Because we're being kicked out at lunchtime.
Oh,
so
okay.
we got
30
some
seconds.
time.
Okay, I'm into the gun. So it was very good. You described the concepts well. You described the relationships between pressure, volume, and temperature, which is exactly what I wanted to see. So I'm glad you brought that up.
Thank you.
Okay, thank you. No, you're fine.
Oh, there we go.
I'm talking about the speakers.
All right, our next presentation is by Brody Stevens, who will be attending NYU Nursing School.
Yeah.
Oh yeah, NYU, very good. So representing NYU, and her mentor was Dr. Kellen Shen.
Okay. Good morning. My name is Brody Stevens, and I'm a second-year student at NYU. I worked in the Gertner Chen lab, and I'm presenting marine models of induced liver fibrosis today. So my initial questions were, what are the most critical cellular and molecular mechanisms driving the progression and regression of liver fibrosis? And what are the key challenges in translating findings from animal models of fibrosis progression to human clinical practice? So just a quick introduction of what liver fibrosis is. Organ fibrosis is a hallmark of disease progression in chronic inflammatory diseases and contributes to about 45% of mortality worldwide. Antifibrotic candidate agents showed promise in animal models, but their effectiveness in clinical trials has been limited and absent. And the progression of fibrotic liver diseases follows shared patterns across liver disease etiologies. And as of right now, there's only one recently approved treatment for liver disease, or liver fibrosis. So liver fibrosis is a progressive accumulation of extracellular matrix, or ECM, that damages the liver structure. It's caused by toxic, metabolic, or viral diseases that damage liver cells and recruit immune cells. HSCs are activated to become collagen-producing myofibroblasts, and typically tissue repair mechanisms balance fibrosis, leading to myofibroblast inactivation or apoptosis and scar resolution after short-term injury. However, in chronic liver diseases, an imbalance between profibrogenic and antifibrogenic mechanisms keep myofibroblasts persistently activated, resulting in excessive ECM production. So liver fibrosis begins with hepatocyte injury, which releases signals activating HSCs. Activated HSCs proliferate and produce extracellular matrix proteins, causing scarring. The Kupffer cells and other immune cells respond to injury by releasing cytokines, which enhances inflammation and fibrosis. And the overall process leads to the accumulation of fibroscar tissue in the liver. So first, I will be introducing the Fibrosis Induction Resolution Project. So the goals and motives for this project were to reduce liver fibrosis using a high-fat diet and validated models representing alcoholic fibrogenesis, non-alcoholic steatohepatitis or non-alcoholic fatty liver disease, and liver fibrosis leading to hepatocellular carcinoma. Then we remove all induction methods, and once the models are established, we track the liver fibrosis resolution to better understand how they spontaneously heal, and we apply these lessons learned to direct human fibrosis therapies. So just a quick display of what this project is. We have the thiocetamide induction model, carbon tetrachloride, and diethylnitrosamine. However, diethylnitrosamine induced more of a liver cancer, and carbon tetrachloride was closer to the human fibrosis model that we were looking for, but thiocetamide produced the most, human replicable model. And those little eyes that are underneath each line are the resolution periods that we have to let the fibrosis resolve. We then collect the livers and then run tests on them. You can see the dosages on the side, and those little question marks are models that we hope to explore in the future. However, today I will be focusing on the thiocetamide model. So thiocetamide is a fibrosis-inducing chemical agent, and it's metabolized in the liver to thiocetamide S oxide. It induces hepatocyte apoptosis and necrosis, which leads to inflammation and activation of those HSCs, which are hallmarks for liver fibrosis. And this chronic administration leads to extensive liver fibrosis. So the methods for this project, we have thiocetamide dosages being 175 milligrams per kilograms 3 times a week, and these mice are injected for 10 weeks starting at age 4 weeks. And then week 10 to week 20 is our resolution timeline. So we can see we're loading up the syringe with the acetamide, which is dissolved in a solvent, and we inject it intraperitoneally into the mice. So looking at some H&E and trichrome staining, this is from a week 8 mouse liver, and we can see that this liver is between stage 3 and 4 Ishak, which is basically just a way of indicating and measuring liver fibrosis, 0 being no fibrosis to 6 being cirrhosis. But it's closer to 4 compared to the carbon tetrachloride, indicated by marked portal-to-portal bridging, which is, if you look at that dark blue and the purple staining, that's basically collagen, like thick fibrous collagen bands. And then we can see hepatocyte death, and yeah, so this is pretty extensive liver fibrosis. And then we can look at the resolution histology. If you notice the thickness of the fibrosis and these bands and the staining, and then by week 19, which you can see closer to the right, this fibrous thick bands kind of start to resolve themselves and thin out, and these huge gaps that we have start to resolve themselves as well. And then looking at some human histology, this is trichrome and H&E staining from a cirrhotic liver at end-stage fibrosis, so it can be categorized closer to stage 4, and when we compare it to our TAA mice, which are straddling stage 3 and 4, we can see a lot of the same histological features. And then looking at the non-viability and like damage of the human liver versus these validated models in the lithotripsy, which I will be talking about later, we can see that they're very comparable, and we find a lot of the same findings. So our preliminary conclusions that we have from this project is that thioacetamide effectively induces liver fibrosis in mice, which is simulating the process seen in human liver diseases, causes notable liver damage, as we see with the histological changes, including fibrosis, necrosis, and altered liver architecture. And it's consistent and reliable for studying the cellular and molecular mechanisms of liver fibrosis. And the next project that I want to talk on behalf of MESM was the non-pharmacological approach using extracorporeal shockwave lithotripsy. So this is a non-chemical way of inducing liver fibrosis. So basically, lithotripters are medical devices that generate ultrasound shockwaves to break kidney stones into fragments non-invasively. And these shockwaves are high-energy sound waves that are focused by a water-filled lens, and when lithotripsy is applied near the liver, the shockwaves can cause direct mechanical injury and fibrosis. And something to remember, and a key and crucial thing to know, is that cavitation is the formation, growth, and collapse of bubbles in a liquid, which can generate significant mechanical forces. And on the right, you can see the lithotripter and what the experiment looks like in real time. So the methods that we use, the lithotripter we use is a Dornier compact sigma to induce this liver fibrosis in C57BL6 mice and diabetic mice as well. The fibrosis and tissue damage was modulated with the utilization of contrast microbubbles and mouse strain, and we also use Optison ultrasound contrast media to provide additional nuclei for shockwaves to collapse. So essentially, increased cavitation equals damaged liver tissue, leading to liver fibrosis. And in this image below, you can see how the vapor bubbles kind of collapse and cause damage to the surface of the liver. And just looking at the features of these livers before and after lithotripsy, we can see discolorization, we can see that the overall shape and nodule of the liver changes as well, and we can know that it works. So the wild type mice histology, this was from a week 10 mouse, and this was a wild type lithotripsy mouse liver. This was classified as Ishak stage 2, indicated by portal fibrosis with or without fibrous septa, indicated by these black arrows. And then the lithotripsy with contrast mouse liver, this was also on wild type, was staged level 3, indicated by portal fibrosis with occasional portal bridging, indicated by these highlighted regions. So you can see that the liver fibrosis is pretty advanced in the mice with contrast as well. And then looking at the diabetic mice histology, it's classified as stage 4, indicated by portal fibrosis with marked portal-to-portal bridging and portal-to-central bridging, indicated by these highlighted regions. So we see that in the diabetic mice, the liver fibrosis is more extensive as well. And then looking at the tissue viability of these wild type and diabetic mice, looking at the healthy Wild type mice, we can see that the non-viability is very sparse. There's, like, the tissue is fine. And then with the lithotripsy between day 0 and day 21, it seems to increase. And then as we add the contrast to the day 0 and day 21 wild type lithotripsy mice, it also increases drastically. And the tissue non-viability increases. And the diabetic mice, it's also the same case. It's a little bit more exaggerated, and we see that as we apply lithotripsy to these diabetic mice, the tissue non-viability increases, and also in the lithotripsy with contrast mice, it increases a little bit more. So some conclusions that we have from this project are that these shock waves, ultrasound, contrast microbubbles, and the differences between the type of mice we use, between wild type and diabetic mice, demonstrate a range of liver fibrosis and tissue damage. The extent of damage increases linearly with the number of cavitation nuclei that become available. So more cavitation nuclei, simply put, equals more bubbles and more tissue damage. The areas of non-viable liver tissue and the extent of liver fibrosis increase throughout the groups, and there's a direct relationship between the cavitation activity and the severity of the tissue damage. And we also know that the diabetic livers are more sensitive to lithotripsy, which translates to say that diabetes exacerbates the effects of cavitation and fibrosis, and they're more prone to damage. And some of the remaining questions that we have with this project are, what effects does lithotripsy with and without contrast have on cell morphology and architecture of wild-type mice liver, and how does lithotripsy with contrast affect diabetic mice differently? So to wrap up my presentation, I feel like my summer has been very fruitful and enlightening within and outside of the lab. I had the chance to get really close with my peers in my lab and also my peers within the program. I spent a lot of time cooking and trying good food in Tucson, and just learning a lot about the mice in general. I got to refine a lot of my skills and also learn a lot more. And I became a hamster mom with 2 beautiful hamsters. But yeah, I just want to say a big thank you to my PI and co-PI, Dr. Gertner and Kellen Chen, and also Messim and Maya for working very closely with them this summer and being great mentors, and also the SIMI staff.
Dr. Chen. Oh, yeah, 30-second comment.
Oh, okay. I already had a question. Yeah, I think this was only one project you worked on. You worked with many others, but I think because we have 3 students, you kind of split it up. But I was really impressed by the amount of work you're able to get done. I'm always happy to see you here every year, so maybe next year also. Saw the picture of you with those hamsters, and I thought maybe those were mice from the lab. So I'm glad to hear that you're that's, yeah. So I was like, is that her in the lab?
No. It was a problematic purchase, but I treat them with love.
Now, this is the 3rd. We can't have any more questions because we're going to be kicked out of this room at a time.
No questions.
But I did want to comment that this was Brody's 3rd experience. She started as a high school student from Central High School.
4th year, and
Okay, in Phoenix. And this year we're giving some special awards in the name of Dr. Walton Van Winkle, who very few people know here. But he was a professor between 69 and 74. And he's the co-author of the first book ever written on wound repair and wound healing by Peacock and Van Winkle. And he was the vice president of Ethicon, which was a subsidiary of Johnson & Johnson. So the fund that we're going to be using, we're giving you a prize of $250 relating to wound repair research.
Okay.
In addition, we're giving Lourdes Castanon, who presented to you, for her work on burns and wounds. We're giving her a $500 prize for her research and care of patients with wounds that wouldn't have healed had she not been in charge of healing them. So I want to congratulate you on that.
Thank you.
And we're happy after so many years. It's been 50 years since he was a professor here, and we're now able to honor him with these special awards. So thank you,
Thank
ladies,
you.
and I hope you come back.
All right, our next student is Liana Tang. She is representing South Point High School here in town, and she was also mentored by Dr. Kellen Chen.
Good morning, everyone. My name is Liana Tang. I'm an incoming junior at South Point Catholic High School, and this summer I was able to continue my medical research and continue exploring my ignorance in Dr. Chen and Dr. Gertner's wound healing, regeneration, and foreign body response lab. After reading the lab's literature, I developed my first question. How can the wound healing process be improved? This is my lab's main research question. Wound healing is a complex and poorly understood process with many faults, including fibrotic healing, hypertrophic healing, with continued worries about the foreign body response. Therefore, the lab's main focus is the prevention and correction of these processes. My second question upon starting at the lab was, how can the wound healing process be replicated in a mouse model considering they heal by contraction? While this is a simple question, I still strove to learn the very basic and fundamentals procedures of the lab. My last question going into the summer was, how can I accept my ignorance? This is a question I've been thinking about since I ended my SIMI experience last year. Last summer, I was given the opportunity to work in Dr. Kenneth Lichty's fetal and regeneration biology lab. Through this lab and through SIMI, I made great progress in truly understanding ignorance and how it works systematically with failure and curiosity. However, I continue to struggle with the acceptance of my own ignorance. So to answer these questions, I was given the opportunity to shadow and participate in 4 different experiments in the lab. The first experiment I shadowed in was an excisional wounding model on high-fat, diabetic, and wild-type mice. So first, the mice were injured with a punch biopsy, and then a silicone ring was sewn around them to promote fibrotic wound healing rather than contractive wound healing. From there, the mice were sacrificed, and their bone marrow, skin, blood, and spleen were taken for processing, sectioning, and imaging for different monocyte counts. This experiment helped me answer my second question, how can the wound healing process be replicated in a mouse model considering they heal contractively? And this is through the silicone ring that was sutured around them that completely eliminated their ability to contract and triggered the mice's fibrotic wound healing process. The next one is a foreign body response experiment. In this case, the contractive style of healing was utilized instead of eliminated. In this experiment, a small wound was made at the bottom of the mice's back. From there, a device was put inside the back, and the mouth was sewn shut. The elasticity of the skin allowed the device to enter the body cavity smoothly without causing much irritation. From there, a foreign body response was induced by placing a mouse in the box, where magnets caused the device to vibrate, which triggered the foreign body response. The third experiment was a breast cancer experiment where tumors were placed in the mouse, where cancer cells were placed in the mouse. The goal of this experiment was to make a reliable and replicable model of murine breast cancer. I did not spend much time with this project, however, I did learn how to section and frozen IF section protocol from this experiment. The last question is a liver fibrosis. The last experiment was a liver fibrosis model. So in this experiment, mice are injected to promote fibrosis, and then the liver samples are taken for sectioning and imaging. Brody just presented on this one too. And then through this, I observed scruffing techniques and orbital blood drawing. Through all 4 of these experiments, I've also made progress in answering the question, how do I accept my ignorance? I've learned to accept my ignorance by asking questions. My lab environment facilitated curiosity as each person was more than willing to help me learn and grow through hands-on experiments and careful explanations. My time in the lab helped me realize the importance of identifying my own ignorance and utilizing my curiosity. I've become a lot more comfortable asking simple questions so I can truly understand what I'm doing and its effects. Moving on to fringe benefits. My SIMI experience has been incredibly beneficial and helped me facilitate my curiosity. My time in the lab has been incredibly hands-on. This offered a nice contrast to last year, where I learned more through shadowing and reading literature. Therefore, I've been able to learn about my ignorance from 2 very different styles of learning. Through this hands-on learning experience, I've learned the how of the lab. I performed my first survival mouse surgery, where I learned the valuable skills of suturing and injections. I've also learned how to necropsy an animal and identify different internal organs of each mouse. On the other hand, I also learned basic skills such as pipetting, calculating dilutions, and sectioning. Another benefit I've taken from this experience is my understanding of an unknown unknown. Through iLog 5, I was introduced to the idea of an unknown unknown. something that someone does not know they do not know. I was able to connect this concept to my time in the lab and my beginning question of how can I accept my ignorance. I've learned so much this summer about concepts I had no previous knowledge of their existence. My time in the lab has been incredibly enlightening of these unknowns, and from this experience, I've made great progress in identifying them. I feel as though I've made significant progress in accepting my own ignorance by acknowledging not only my known unknowns, but my unknown unknowns. These are my final questions. Firstly, there's the question that the lab is answering as a whole. How can the wound healing process be perfected? This summer, I've learned that the health effects from the wound healing process can be harmful and even deadly. To answer this question, I will continue my laboratory research over the school year in Dr. Chen and Dr. Gertner's lab. This summer, I also focus mainly on fibrosis. Fibrosis, the creation of scar tissue, is how the human body heals after wounding. However, neonatal and fetal humans heal through regeneration, and therefore the goal is to find a way to promote regenerative healing. And the last question is, how can I utilize my ignorance? While I've made significant progress in understanding and accepting my ignorance, I feel as though I still struggle with utilizing my ignorance to improve. I would like to end this presentation with a quote from Chinese philosopher Confucius. Real knowledge is to know the extent of one's ignorance. Ignorance is dangerous, but once accepted, it can become a tool and the driving force behind curiosity and therefore science as a whole. Thank you so much, and are there any questions?
Ellen, do you have a comment, 30 seconds, and then if there's a student, I'd like to ask a student a question.
Yeah, Leon, I think you did a great job this summer. Clearly, I think your presentation nicely showcases a high level of sort of the different experiments going on in the lab. And I think one thing to think about too is, we have these different mouse models and how we try to leverage both the pros and cons of, animal research and these different models. I think there's different sort of assumptions you have to make and different sort of sacrifices. And I think, since we're going to continue to work together, that last slide was interesting because last year I think we worked in a fetal regenerative lab with Dr. Lichty, and that's something we kind of think about for wound healing in our lab, but sort of don't have as much hands on it. So maybe a way for you to leverage both of your SIMI experiences, you can start asking some questions about, what are some similarities between the labs and sort of how can we, fuse sort of our collaboration to improve the lab that way. So congrats on a good summer.
Thank you.
From the audience, 30-second question. Terry, do you have a 10-second question or a comment, a word?
Just real quickly, fibrosis is great, wound healing is great, but sometimes your body heals too much. For example, when you get a heart attack or if you have fibrosis,
Right.
is there ways to fix that or control
Well,
that?
that's what they're working on.
Yeah,
That's what they're working on. Okay, got it. Okay, thank you.
Don't steal the next few years and summer and thunder. Okay.
Thank you.
All right, our next student is Olivia Rencher. She is coming to us from Marquette University, and she is also in the Kellen Chen lab.
Hi, you guys. My name is Olivia. You got a pretty good introduction of our lab, I think, from the first, the second and third student that presented. But the main question I was trying to answer this summer was, what role do Ly6C2 and CD64 monocytes play in diabetic wound healing? So I feel like we first need to understand why we care about answering this question. So diabetics have been proven to heal much slower in a much more prolonged phase of healing than a non-diabetic patient. So first we need to understand why that's happening. Also, as you can see in that image, inflammation is the second stage in the 4 stages of wound healing. However, diabetics have shown to be in a chronic inflammatory state, which may cause them to be constantly recruiting these leukocytes or other cells to basically respond to this inflammation, and that could be inhibiting or delaying their response to normal wound healing. Boop. Boop. What? Okay. Is that my second slide? Yes. Okay. So First, we need to understand what we're actually looking at in the blood and tissues of these mice to study these different cell populations. So in general, leukocytes are these white blood cells recruited to respond to injury in order to reduce inflammation or prevent the risk of bacteria from causing disease and infection. So you can divide these leukocytes into two different classes, one being myeloid cells and the other being lymphoid cells. We want to focus on myeloid cells specifically because they are innate responders that respond immediately to injury, which would make sense if we're trying to do fast-acting wound treatment. So specifically, we want to focus on these monocytes, as research has proven that monocytes are highly expressed throughout the entire wound healing process, proving their significance somewhat in wound healing. So, oh my god, I double skipped again. So we specifically want to focus on Ly6C2 and CD64 monocyte populations. From our previous research collected by Katarina Fisher, we found that these Ly6C2 monocyte populations were highly expressed during the wound healing process of our wild type mice, but they were not highly expressed in our high fat model, which made us curious to say, maybe that means something. Maybe that's causing this delayed response because these monocytes aren't as highly expressed in these high fat diet models. We did notice also that there was a pro-inflammatory cell population during diabetic wound healing, different than the monocytes observed in our wild type model. So from that, we decided to target our Ly6C2 monocytes as well as the CD64 monocytes, as that has been a proven marker of inflammation. So methods. So we are using C57BL6 wild type mice, also creating two groups with just our control wild type model and then inducing a high fat diet to create a diabetic mouse model. So as Liana had said earlier, mice heal differently than humans. So in order to inhibit their biological response, we need to suture a silicone ring around the incision that we had made, holding that skin in place to basically mimic like a human tissue reconstitution response. So we'll do excisional wounding while they're under anesthesia, do subcutaneous injections prior to surgery for pain. So for mice that have undergone surgery, going through the process, we will do dressing changes basically to visually observe how that wound is healing and perform tracings. We will also do antibiotic administration if the wound looks infected. So sample collection is what we do on days we are sacrificing the mice to collect bone marrow, spleen, blood, and skin in order to run flow cytometry as seen in that graph. That's not our specific data, but that's just an example of kind of the flow cytometry and gating that we do. Basically analyzing the height and area in a linear display to see like basically if the cell is like healthy and alive, to make sure we're actually studying live cell populations, not dead and throbbing cell populations. And then we'll do, like I said, data collection, which is we'll do staining and imaging, and then we'll also do flow cytometry to see what monocyte populations are present during their healing, and contrast that between the wild type and diabetic mice. So some fringe benefits that I have obtained through the summer was definitely learning to anesthetize the mice for survival surgery procedures, learning to perform sutures on live mice following excisional wounding, learning to deliver pain medications via subcutaneous injections, learning to process different tissue samples into single cell suspensions and interpret those cell populations displayed on that flow data. I also was lucky enough to get to help Messam on his liver fibrosis project with Brody, so I was able to learn how to collect blood retroorbitally and handle the animals to scruff and give retroperitoneal injections. So in doing this research, we found an inverse trend shared between the wild type and diabetic mice, seeing that after the day of incision, the DB mice, their monocyte populations drastically declined, whereas it was the opposite trend for wild type mice, with a major statistical significance from day 0 to day 2, as you can see on that chart for our wild type mice. So clearly it's almost immediately responding to that injury, whereas right when they're injured, it seems they start to decline in the DB mice. So what we can say from this data is that there are high concentrations of monocytes in DB mice prior to and on the initial day of wounding, meaning that a high-fat diet induced diabetes upregulates these Ly6C, CD64 monocytes and then also we can say that there's a significant increase of Ly62, CD64 monocytes from POD0 to POD7 starting on day 2 even for these wild-type mice. So some conclusions we've drawn this summer was consistent and changing expression of Ly6C2, CD64 monocytes throughout healing, proving significance in wound healing process. Wild-type mice express high concentrations of Ly62, CD64 monocytes 2 days following excisional wounding, and DB mice display a higher populations of Ly6C, CD64 monocytes prior to excisional wounding. Some questions still remain left unanswered. Is this downward trend of monocyte populations seen from postoperative day 0 to postoperative day 7 in DB mice caused by external factors that we have not yet studied? Why is there an inverse trend shared between diabetic and wild-type mice from postoperative day 0 to postoperative day 2 and even to day 7? And does this support our assumptions? And then this final question is kind of framing where we're going next in our research, which is by increasing the populations of Ly62, CD64 monocytes, is a wounded diabetic patient. Would this enhance their delayed stages of tissue repair? So in order to do so, Maya and Dr. Chen have talked about manipulating these monocyte populations in the blood of diabetic wounded mice to see if that enhances or basically delays their process even further of healing. This could make major advancements in the future if we see positive results. Oh. Thank you. Questions?
Any questions? Give you 30 seconds. Kellen, do you want to comment?
Sure, yeah. I think this is a good showcase. I mean, there's a short amount of time. I've talked about some cell populations we're interested in. Obviously, there's a lot of time spent on a lot of groups and things are kind of changing up and down. I think you sold yourself short, though, on some of the good work you've done over the summer, because I think there's been, we've been basically having to collect skin and blood and different tissue from these different mice at different time points, and also doing like N=10 wounds for each of these mice. So there's been a lot of good sort of sample processing that you've been involved with that I think has really contributed to, the next directions for our lab. We're doing a lot of flow cytometry now, which we were sort of learning over the past year, and I think you've come at a good time, and that's been sort of going pretty rampant. So, yeah, I think it was a good summary, and congrats on a good summer.
Thank you.
All right, our next student is Leilani Salazar. She's coming to us from Douglas High School, and her mentor is Christine Hoyer Kimura.
Good morning, everyone. My name is Leilani, and I'm a rising senior at Douglas High School. I work with Dr. Christina Hoyer Kimura this summer, and today I will be presenting my presentation that is based off of my experiment of P95 decreasing microglia activation in heart failure. So, heart disease is the leading cause of death in the United States. There is a large population of individuals who have heart failure, and of these individuals, many will end up developing cognitive impairments as well. It is stated that 75% of people with heart failure develop cognitive impairment, and this image is an example of how heart failure involves cerebral perfusion and impaired audio regulation and systemic inflammation, which then leads to issues that cause changes in the brain, such as cognitive impairments and anatomical issues. So in Dr. Hayes' lab, we investigate a treatment that prevents inflammation in heart failure associated with developing cognitive impairments such as microglia. So microglia are immune cells in the brain that help maintain it to be healthy and are responsible for multiple functions. Microglia activation can look different depending on their structure, appearance, and functional state that the cells are in. So as you can see in the first image, microglia can appear different in different states, such as hyperramified microglia, activated, amoeboid, rod, and ramified, and they can either go to one state in that order, or they can jump from another state, such as activated microglia can become ramified, hyperramified can become rod, amoeboid can become ramified, etc. And then the next image to it shows what resting microglia looks like. And resting microglia shows that the cell branches are long and uneven and are spread out in different directions. And as microglia becomes activated, the cell branches change to become shorter, thicker, and are hyperramified, which then the cells eventually develop into an amoeboid shape, which is at the end of the image right there. So overactivated microglia can kill neurons and lead to cognitive impairment. So in order to prevent and reduce heart failure and cognitive impairments, a therapeutic drug known as PNA5 is being studied to see whether it is safe and effective to help people who suffer from these health issues. So PNA5 rescues cognitive impairment and heart failure. In Dr. Hayes' lab, a study from the past has shown that PNA5 reverses cognitive impairment and dementia that is induced by chronic heart failure. The results from that study showed that mice models with heart failure given PNA5 had shown improved cognitive abilities. So the first image shows a novel object recognition test, which is basically a test to see how well mice's cognitive function and memory work. So as you can see, on the y-axis there is the discrimination ratio, and on the x-axis there are three different groups of mice. So the two bars above the line labeled as control and VCID and PNA5 are the mice groups that showed a positive discrimination ratio, meaning that their cognitive function and memory was good. However, the bar below the line labeled as VCID vehicle were the group of mice that showed a negative discrimination ratio, meaning that their cognitive function and memory weren't as well as the other two groups. So all the mice were then treated with PNA5, which then reversed the neuropathological effects of vascular cognitive impairment and dementia and heart failure. So this suggests that PNA5 could be a possible effective disease-modifying therapy to treat and prevent vascular cognitive impairment and dementia. And then the second image shows the structure of PNA5, and what PNA5 is basically a glycosylated angiotensin 1-7 that works by activating the mast receptor agonist on cells in the brain that include microglia. So in Dr. Hayes' lab, our hypothesis is that PNA5 will decrease vascular cognitive impairment and dementia and heart failure associated microglial number and activation in the CA1 and C3 region of the hippocampus. In our lab, we also aim for two things. The first one being to elucidate microglial response to heart failure, and the second one is to determine PNA5's ability to reverse heart failure associated microglial response. Moving on to the methods, in the study, multiple methods had been performed and materials were used. So as you can see, there were three groups of mice that were used, and they were the Shan saline mice, the heart failure saline mice, and the heart failure PNA5 mice. The first image shows a timeline of the surgeries and treatments that the mice had undergone from 0 weeks to 8 weeks, and what's most important to know is that only male mice were used in this experiment. So by the end of 8 weeks, their brains were collected for an immunostaining for microglia, which was done by using the IDA1 marker, which stands for ionized binary calcium adapter, which detects microglia. So pieces of tissue of the brain were put on a slide. They were then stained with IDA1, and then they were put under a microscope, and under that microscope, activated microglia was seen. So then another process that was used was the process of ImageJ, which basically showed images of the CA1, C3 regions of the hippocampus to detect and count any microglial activated cells. So once the cells were counted, they were then skeletonized, also under ImageJ, to measure the lengths of the branches of the cells that had been counted and were activated. So my results were inconclusive, being that there was no significance across the groups and more mice were probably needed to identify any trends. But as you can see from the images, these are the CA1 and CA3 regions of the hippocampus, and it shows the cells per area for the 3 groups of mice that were used. And under those images, you can see the activated cell counts of microglia, and they're pretty visible to the eye, being how big they are as well. So once again, my results were inconclusive, and if in order to be significant and conclusive, then there probably would need to be a larger group of animals. And it also appears that heart failure is different than both PNA5 treatments and the control groups. And there are more images of CA1 region and CA3 region that show the average branch lengths per cell. And then at the bottom, there are cell counted images for the 3 groups. There's the Shan-CV mice, the heart failure vehicle mice, and the heart failure PNA5 mice. So to conclude this presentation, while the results of the experiment are inconclusive, PNA5 treatments could possibly decrease microglial activation and vascular cognitive impairment in dementia and heart failure. Similar to my results, the results from the lab's previous publication showed that mice models of vascular cognitive impairment and dementia and heart failure had shown an increase in microglial activation in the CA1 and CA3 region of the hippocampus. The differences in my results compared to the previous study are likely due to how I had a smaller group of animals to work with, and also how I could gradually improve the techniques that I used during this experiment. So to be able to explore longer effects of PNA5 treatments, clinical trials should be performed to help and reverse the effects that a patient with vascular cognitive impairment and dementia and heart failure experiences. However, there are certain things that could be more explored in the study and answers such as, are there sex differences between microglial activation and the mechanisms of inflammation in males and females? And since only male mice were treated in this experiment, will there be any different responses when testing the female mice with the treatment of PNA5? And for my acknowledgements, I would most want to acknowledge my advisors, being the Hayes Lab, Dr. Meredith Hay, and my amazing mentor, Dr. Christina Hoyer Kimura, as well as the people who are in charge of the SIMI program.
Okay, I'm going to ask you for your remaining question. Not a direction, not a hypothesis, not a conclusion. What is your remaining question? With a question mark at the end of it. You had so many this summer.
Does it have to be based off my experiment? My probably main question is, since only male mice were tested in this experiment, will there be any different responses when testing the female mice with the treatment of PNA5?
With a question mark at the end of it, right? Okay. Now, I will always.
All right, our next student is Dea Kochan from Bases Tucson North, and she was mentored by Dr. Woolman.
Hi everyone, so my name is Dea Kochan, and I'm going into my senior year at Basis, and I had the pleasure of working with Dr. Lila Woolman and everyone else in the Fregosi Woolman lab this summer. And today I'm going to be talking about my SIMI experience, which can be honestly summed up in a phrase: curiosity and nicotine. So to start off, I'm going to be talking about my lab a little and giving everyone a better idea of what I've been working on. And then I'm going to dive into my starting questions and the progress I made in answering all of my questions. And then I'm going to say what I still have left to work on and how I plan to explore these questions, and then a time to just thank everyone. And so to start off, my lab had to mainly do with how chronic nicotine exposure impacts breathing control and respiratory rates in mice, especially in hypoxic conditions. So for our lab, we had, or my main lab I should say, that we had 3 groups. We had a control group, a nicotine exposed group, and a nicotine withdrawal group. And so all the rats had a tracheostomy performed, and we also took blood samples from the femoral artery. And with that, we collected all the blood samples and measured their breathing rates, and we found out that in the end that the control and the nicotine exposed rats showed no difference when exposed to hypoxic conditions. They all were subjected to 3 levels of hypoxia: 15% oxygen, 12% oxygen, and 10% oxygen. And we found that while the nicotine exposed and the control showed no statistical significant differences, that our nicotine withdrawal rats, who were timed to place like a day of withdrawal, showed clear signs of hypoventilation, and we found that there were sex differences as well between only the nicotine withdrawal group. Even though both showed signs of hypoventilation, from the blood samples we were able to see that the male nicotine withdrawals had a normal pH and high bicarbonate levels, whereas the females had a normal bicarbonate level but low pH, which means that the male nicotine withdrawals were in fact compensating for their hypoventilation by having their kidneys retain bicarbonate. And even though this was my main lab, I had the pleasure of being involved in many different labs and many different techniques. So for example, this picture here on the right is a different experiment that we were working on with nicotine exposed neonates, and we would take out the spinal cord here and the medulla, and we would take the central nervous system recordings from specifically the C3, C4 branch, which sends recordings to the phrenic nerve, which controls the diaphragm, and we would expose them to either a nicotine, a strychnine, or a cotinine bath and see how the signals change. So to get into my starting questions, the big one, especially with my main lab, was how does nicotine withdrawal impact daily function? Another one was why do some rats go under anesthesia quicker than others? This was noticeable in most of my experiments where the rats would sometimes take 2 minutes to go under, and other times, as Dr. Woolman well knows, around almost half an hour. Another thing was how some rats can still breathe normally after going under a phrenicotomy, which is when you sever the phrenic nerve to paralyze half of the diaphragm. And another one that I got from the experiment I mentioned right before with the neonates was why the signals from the central nervous system will randomly spike before going down to the average amplitude seen from the other recordings. So to get into the progress, I feel that unfortunately I did not have as much progress as I desired, and I feel like this is due mainly to the fact that I was being a part of many different labs, and yet I had less time to work on some of these other labs where some of my other questions stemmed from. But I did figure out specifically in regards to nicotine withdrawals impact on daily function, specifically with breathing, that as we saw that nicotine withdrawal can severely impact breathing control by causing extreme hypoventilation in intensive conditions such as hypoxia. And we also found out that rats can in fact breathe normally after undergoing a phrenicotomy, even though half of the diaphragm is paralyzed. Somehow the lungs are still able to compensate even in extreme conditions. So moving on, for my fringe benefits, I feel that overall my experience at SIMI was very positive, and lab experience was something big for me this year. I feel that even though perhaps it was not as hands-on as I would have liked, that I was exposed to a lot of variety of techniques and a lot of different experiments, and I was also exposed to all different phases of the research pathways, from preliminary data to grant writing, and I also got a lot of advice from the people in my lab. We're a pretty small lab, and I feel that allowed me to make connections with a lot of the people in my lab, and they gave me a variety of advice ranging from college, furthering my education, the research field, and some general life advice. Also, I feel that this program has really cemented my passion for research. A big reason that I applied for this program was to see if the research field would be something that I was interested in, and I feel that after this I can concretely say that research is a field you're good, that research is a field that I wouldn't mind working in. And then of course, mentorship and networking. Dr. Woolman was an amazing mentor, and she exhibited all of the key qualities I feel that a good mentor should have, and she was always very patient with me and always did her best to explain everything and really pushed me to be someone who was more independent and someone who wasn't going to shy away from asking when they didn't understand or just asking if they can get involved with something. And of course, networking. Dr. Woolman didn't only just show me all the other people in my lab and in this building, she let me join in her journal club meetings, and she also helped me set some ground connections from people not just in America but from all around the globe, including Brazil. So my remaining questions is, I still haven't figured out why the signals from the central nervous system can arbitrarily spike. And of course, the big one coming out of my main lab was why female rats who were nicotine withdrawal rats didn't compensate for hypoventilation, and also what sort of complications can occur during a surgery or in recovery from long-term nicotine use. I feel though actually with my third one that there is a partial answer here, especially with the in recovery, because a main part of our lab and sort of the clinical implications of this is when someone undergoes surgery and they have consumed nicotine for excessive periods of time, going into surgery places them in immediate nicotine withdrawal, which can hamper their recovery in the future, and especially as we've seen with the experiments where the withdrawal sent them into hypoventilation. And so how to proceed, I feel that I should start small and maybe pick one big question and start to just break it down into key components, because Dr. Woody taught us to keep our questions broad, but I feel as a start I should start to break them down. And I would also like to join a lab during this school year. Unfortunately, I can't continue in Dr. Woolman's lab as she's sadly leaving, but I would still like to work with someone else, perhaps in the neurophysiology department. And hopefully, if Dr. Woody allows it, I can apply for SIMI again, perhaps in my undergrad year. So I just want to thank everyone for listening to my presentation, and I just want to thank everyone in my lab, and of course all the SIMI staff for crafting a very positive experience for me this summer.
Some comments.
You saw a really wide variety of things in the lab that you described. So it's really impressive that you were able to talk in such detail about each of those things, considering that you worked with adult rats and you worked with neonatal rats, and all of the experiments dealt with nicotine, but the perinatal nicotine exposure and adulthood exposure questions were so different. So you really did a good job of understanding a very broad range of topics.
Any other questions? Enjoy the renewal.
All right, for our next presentation, we actually have a duo presenting together, and that is Dylan Brashini Avia from Yuma Catholic High School and Amaya Noriega from here at University of Arizona, and they were both mentored by Dr. Sweeto.
Hello, my name is Maya. Like Lisa, thank you. This summer we were in Dr. Cohen's lab, and to summarize it, we were doing research in menopause and the microbiome.
My name is Eileen, as Kino said, and we are under the mentorship of Dr. Sweeto.
Okay, so to begin our starting questions for me were, what makes the microbiome in our gut want to live there, and why do they want to live there, and how do they live there? And then another question was, what creates a good diet? Is obesity naturally occurring if it can happen with changes in hormones?
And so my starting questions were, what is the gut-brain axis? How much does it impact our decision and body? How do you test for cognitive regression? How do you test for cognitive regression? To give some background into our research, we wanted to give a definition of what exactly is menopause for people who don't really know. Menopause is the discontinuation of estrogen, or when a woman stops menstruating. There are 4 types of estrogen: E1, E2, E3, and E4. E2 is essentially when is the primary hormone when it's like in a woman's reproductive years, and it basically handles the reproductive functions. E1 is the estrogen during menopause when it is released. E3 is the estrogen that is released during pregnancy, and E4 is the natural estrogen when you develop as a fetus. Since it is a very critical hormone, it also has cardioprotective agencies. So when you essentially have a decrease in estrogen, it leads to diseases and conditions such as obesity, cardioregressive diseases, obesity, and osteoporosis. The way how we research this topic is by using an occupational chemical known as VCD, or 4-vinylcyclohexene dioxide. We inject this into mice, where it essentially induces menopause by disrupting the primordial and primary follicles that produce the egg during menstruation, and so we are able to study its processes.
So another point to get back on is the microbiome. So the microbiome is basically what's happening inside your stomach and your intestines. It's composed of different bacteria that lives in there, and this bacteria is known as healthy bacteria, and good bacteria is beneficial. So when you eat something and they say you're eating probiotics, that is the good bacteria that's inside of your stomach. That is what you call your gut flora. And then something that we can find inside of our gut is an enzyme called beta-glucuronidase. It is an enzyme that acts as a way to dispose of stuff within our system. So what we're trying to do in our lab is trying to figure out how to use that enzyme to keep estrogen within the body, which can further keep the women protected from other cardiovascular and issues that can arise from menopause. So our lab primarily has 3 different focuses, mainly because there are different master's students and undergrad and PhD students working there as well. So one of the main PhD students is Vito, and then our postdoc Sparky, who they were studying the intestinal stem cells derived from menopausal mice and the implications for screening cardioprotective probiotic species.
And also for our 2 master students, Ryan and Shelby, Ryan views the ability of probiotics to reduce damage from heart attack and menopause by measuring the B-GUS or beta-glucuronidase in fecal matter. And Shelby determines the correlation between menopause and heart attack damage and cardiovascular issues. So for our research and contribution to our lab, we both have our individual experiments. We look at how they kind of connect to the microbiome and to different cognitive conditions. And we see the relation between the 2. I had a focus on Parkinson's, and Maya had a focus on Alzheimer's, and we see its contribution of estrogen to the gut-brain axis, which is essentially the two-way communication between the gut and the brain, and it sends signals vice versa. Like if you're nervous, the brain sends signals to your stomach, and that's kind of when you feel that like gut feeling, kind of when we're presenting right now. And that's essentially what we did. We had a 4 in their laboratory testing. We had 4-MU, which is kind of like just what we use hydrogels for, hyaluronic acid essentially. And we use that as a mock sample, and we tested that to see its effects and how it kind of contrasts to control samples to see its intestinal enzymes. And we also researched different treatments, such as using hormone secretion to we implant it or inject it as we did into the mice. We inject it into their intestinal microbiome, and to trigger the gut-brain axis, as we said.
So some other parts of our research that may be less known by people who, or that should be emphasized just as much, is a journal club, career exploration, and the overall development within our lab techniques as well as our lab knowledge. So journal club, basically, if you don't know what that is when you gather around other people around you and you explain research articles that are relating to your topic. So for example, we participated in a few journal clubs. One of them was where we talked about VCD, which is the main component that induces menopause within mice. And we were able to talk about what that means within a relationship with our lab, how it actually works within the mice, what it actually does, what it looks like, and how that, and just how to use those articles to give us a better understanding and for all of us to be able to know what's happening within the lab and what we're doing with those different types of chemicals. And then for our career development, we were able to be given mentorship, like we said, by Sparky and by others around us, to be able to give us insight in what future plans will look like and how we should plan ahead, and as well as giving us other opportunities to talk to people that are within the fields that we are interested in. And then we were also able to develop our skills within the lab. As we said, we refined our pipetting skills as well as learning how to read plates. We perfected, I believe, Excel, which I believe is a difficult feat anyways. And then we were able to further develop our skills within lab etiquette as well as being able to see the works and the ins and outs of the lab that we were in.
So the kind of overarching question in our whole lab is, how can we help women that are suffering from menopause and its effects, and more specifically in our own projects, how can menopause and its consequencing conditions be affected by the gut microbiome? So some of the goals we hope to reach through these questions are to help menopausal women's resulting conditions, research and develop knowledge for women's health, and learn how to use the microbiome to help developing conditions. Some of the goals we hope to reach, it's important to attempt through these goals. It's important to attempt these goals because women are all around us. A lot of the women face this feat at some point. So it's really important to bring this to attention. Also to the fact that women's health is a typically under-researched topic. So being able to make this more widespread in the research field is really important. The loss of estrogen, as we said, leads to dangers to their daily lives and introduces diseases that severely impact how their lifestyle is. So it's important to look at that.
So for the data we said, in order to be able to get the skills that we did in the lab, we did different protein assays and quantifications. So protein assays determine the concentration of protein. So as you can see right there in this 96 well plate, it shows a gradient from blue to this green. So I believe the higher the concentration, the more blue it's going to be, and the less the concentration, the more brown it's going to be. And then the way that it looks like that is you can see the dilutions on the first 3 rows from A. There's laser pointing here. Yes. Okay. So these first 3 rows right here are going to be our known standard samples, which are what we use to create standard curve. And then right here we can see our unknown samples, in which we'll use our standard curve table to find out what these unknown concentrations are based off their absorbency. And then right here you can see the samples that we use for other quantifications, which measures the enzymatic reaction and activity within a sample. So the protein assays, this one is different from the quantifications, but it still equally gives just as much as information and is important in lab data. So from analysis, as David mentioned, we have our flora and new samples here. This is one of the graphs that we created. It gives us our equation, which does help us find the overall concentration from our unknown samples. And then over here, we can see another type of graph that was given to us by our mentor, as well as the data that we collected. So the orange is what you can see from Sparky's data, which is a mentor, and then the blue is from our data. We can, from here, determine also what the concentration is of our unknown samples. So for our remaining questions, my questions are, since inflammation is the cause of many different types of conditions, can we use the microbiome to be able to treat different types of conditions in the brain as an alternative route to the blood-brain barrier? And how would we use this how would this experiment work with women who have one ovary versus two ovaries, or have prior issues within their ovaries that would hinder the full process of menopause or provide other different types of side effects from not experiencing a traditional route to menopause?
And some of my remaining questions are, how can different treatments for menopausal relief affect the woman's psyche, as we know that some of these side effects can be pretty detrimental? And how can we fix those treatments to reduce the negative side effects? Another one is, how, after wax embedding intestinal tissue, what is the next process to help discover B-Gus activity in the microbiome? Since in our lab we also did wax embedding processes to help study further as the lab progresses. Unfortunately, we are not able to view those processes after the wax embedding. So that's just my remaining question after this program.
So these are some of our fringe benefits. As you can see, we have our lab people right here. This is pretty much almost everybody in our lab except for Shelby, who's another one of the master students. This is them preparing for the wax embedding, which you can see right here. This is the microtome, which is after they create these little wax figures are what the wax looks like. When they melt it, they dip the samples that they collected from the mice during surgery, and then from there it gets cut into these very, very thin slices where you can put them onto a microscope, and then you can see what they look like under the microscope. Here you can see what the bacteria, this is the bacteria that creates food, or the food inside of your intestines, I believe, or for the other cells inside your intestines. This is what they look like under the microscope. Here is an overall picture of our lab. Here is us collecting some of our data from when we ran our plates. Here is our mentor Sparky showing us how to do our calculations, as well as get a better perspective of what happens between the data side of science. And this is her doing this part where we generate the food and extra. This is just the extra cells from this, but it's still neat to see that they're all in this little cup right here.
Yes, and some of my fringe benefits were mainly getting a lot of exposure to what a laboratory looks like and how professional research is conducted, which is something I had never seen before. I had initially thought that I only wanted to do clinical work, but after seeing how research is conducted in the laboratory, it definitely exposed that pathway for me and is very, very tempting to pursue. And also just kind of seeing definitely new equipment I had never knew existed, so that was pretty neat. And also having a lot of resources nearby and a lot of people nearby that could give me a bunch of advice. Like Amaya here, she gave me a bunch of advice about U of A and the campus and what kind of life looks like for me as an undergrad, as a future undergrad coming here for my undergrad studies. So thank you. Any questions?
Yes, so I had the privilege of working with Galen and Amaya this summer. It was absolutely fantastic. It's really common when people are exposed to things, to difficult concepts they've never seen before, difficult tasks to kind of freeze. And I was extremely impressed that that was not the case for either of them. We did a lot of difficult concepts. We did a lot of various things. A lot of the concepts that I forced Dylan and Amaya to work with on curricular pieces were things that you might actually not really have to deal with unless you were like a grad student in a research lab, and they were both really game for going for it, and they did really well. I was also impressed with like curiosity that they showed and kind of like everything that they were exposed to, and the self-motivated self-paced learning that they did. There were also like the concepts we work with in the lab are really difficult. I've been in the lab for about 8 months now, and I'm still a lot of it is still new to me, and so they were also there, as they mentioned, there are multiple
All right, our next student is Vic Sangster-Vai, and he is coming to us from here at University of Arizona, and he was mentored by Dr. Pires.
Yes, my name is Victor Sangster Biget. I am an incoming senior here at the University of Arizona, studying physiology and medical sciences. This summer, I had the opportunity to analyze the morphology of the mesenteric artery in 5XFAD Alzheimer's mouse model. So this summer, I worked in Dr. Pires' lab. Dr. Pires is an associate professor here at the College of Medicine under the Department of Physiology. Dr. Pires' lab focuses on understanding the vascular physiology under disease conditions such as Alzheimer's disease. And while doing this, we also explore possible therapeutic targets. So Alzheimer's disease is a neurodegenerative condition that causes cognitive impairment, and a general characteristic of this disease is that is associated with the accumulation of amyloid beta plaques in neurofibrillary tangles. Certain age-related vascular alterations can aid the progression of Alzheimer's disease. So examples include atherosclerosis, microvascular pathology, and hypertension. There are currently no long-term treatments available for Alzheimer's disease, so it is important to study this disease. And to study this disease, we use the 5XFAD Alzheimer's model. The 5XFAD Alzheimer's model is a transgenic mouse. Therefore, there are 5 genetic mutations to the amyloid beta precursor protein as well as the presenilin-1. And these gene mutations cause the mouse to have familial Alzheimer's disease, which is considered early onset. And we also use this model because it is difficult to obtain human tissue, and it is also easier, and it takes less time for cognitive impairment to occur, which approximates to 4 months. So the overall research question on this project that my mentor and I are exploring is vascular dysfunction present on the mesenteric artery in Alzheimer's model 5XFAD. So the mesenteric artery is a peripheral artery, which allows us to focus on peripheral resistance. And we generally collect the second branch of the mesenteric artery, and the reason why it is considered a peripheral artery is because it has a lumen diameter of less than 200 micrometers. And arteries generally have a contractile component, and when this contractile component is disrupted or affected, it leads to vascular dysfunction. And there are 2 possible explanations to this that we can explore, which is the production of vasoactive molecules and structural changes or modifications. And to do this, I apologize. My question over the summer was, does vascular remodeling occur in the mesenteric arteries of 5XFAD mice after cognitive impairment? And to answer this question, we used hematoxylin and eosin staining. Hematoxylin is a stain that stains for the cell nuclei. It generally appears under bright field microscopy with a purple hue, whereas eosin staining appears pink and stains for the extracellular matrix as well as the cytoplasm. And this allows us to image our tissue, and after imaging, we then explore these 4 parameters, which are the lumen area of the vessel, the vessel area, the vessel wall area and a ratio that compares the lumen area and vessel wall area. And that is shown with this equation. And I must mention that this ratio is an index for vascular remodeling. So, to the picture on the right, there are traces in yellow and red. The yellow is the lumen area, whereas the vessel area is red. And to obtain values for the vessel wall area, we have to take the difference between the vessel area and the lumen area, which will then help us obtain a value for the ratio. So, getting into what we observed, there were no significant changes in the lumen area when comparing these two groups as represented by the graph and the images. There were also no change in the vessel area. There were no changes in the vessel wall area between these groups as well. And considering that there were no changes in these previous three parameters, there are no changes in the lumen area vessel wall area ratio. So in conclusion, because there are no changes to the lumen area, vessel wall area, as well as the ratio, this information allows us to explore the production of vasoactive molecules to better understand how peripheral vascular dysfunction presents in 5XFAD. To officially form that into a question as a remaining question, that brings us to exploring what other possible mechanisms explain how vascular dysfunction presents in Alzheimer's disease. So, some fringe benefits that I obtained over the summer include gaining lab experience, as well as analyzing scientific mindset to explore open-minded curiosity, as well as obtaining lab skills such as being more organized, being analytical, strategic, and open-minded, as well as learning new lab techniques and networking and mentorship. I would say receiving and giving mentorship to from my mentor as well as to high school students. So the use of ILONs over the summer definitely helped me explore this concept of open-minded curiosity. And lastly, I would like to acknowledge Dr. Paulo Pires for having me in the lab, as well as Dr. Josiane Fernandez de Silva for working with me, and Dr. Marlise Ritty for having me in the SIMI program, and as well as Grace Wagner and the rest of the SIMI staff. Would anyone in the audience have a question?
It started out in high school. Was it Globe High School where you were sophomore?
Yeah, I was.
He was one of the youngest.
I was entering my
high school.
junior year. Globe High School. Yeah.
And he's worked in the fire lab during that time. Is Paolo here? Or him from his lab?
Thank you, I appreciate that very much.
Any questions from the audience? I think this room will not even hear the sound of the rooms downstairs.
All right, our next student is Akash Kumar. He's coming to us from Basis Oro Valley, and he was also mentored by Dr. Pires.
Hi everyone, my name is Akash Kumar. I'm going to be a senior at Basis Oro Valley, and this summer I had the opportunity to work in the SIMI program with Dr. Paula Pires. And my project was on the effect of anti-amyloid beta antibody on amyloid plaque in TG-SWDI mice. And a little bit of background on Alzheimer's disease. Alzheimer's is a neurodegenerative disorder that affects approximately 47 million people as estimated in 2015. And there are projections saying that the number is going to be about 131 million people in 2050. And Alzheimer's patients tend to display a cerebral vascular damage, which leads to an accelerated progression of Alzheimer's disease. ARIAs. ARIAs are amyloid-related imaging abnormalities such as edemas or hemorrhages, and they can occur in Alzheimer's patients who are receiving immunotherapy. And it's important to reduce ARIAs on Alzheimer's patients receiving immunotherapy by learning the mechanisms that result in ARIAs. So if you look on this picture over here, this little white area would be a hemorrhage, along with this area right here, these are hemorrhages on MRIs. Now one theory about why this happens would be if we were to look on top, right here is a healthy vessel where the vessel is able to siphon out amyloid beta quickly enough. However, in an impaired vessel, it would not be able to do so fast enough, resulting in a plaque buildup, which would result in damage to the vessel, resulting in a hemorrhage. Now, what questions did I begin with? Does the immunotherapy reduce amyloid beta plaque? Does PNA5 improve the immunotherapy? Does PNA5 affect amyloid beta plaque content? And how I went about answering this was I used TG-SWDI mice, which is a transgenic mouse, and I used anti-amyloid beta antibody therapy, which is IgG, as well as PNA5, which is an antioxidant and anti-inflammatory drug. And I had 4 experimental groups. I had my vehicle and my PBS. I had a vehicle and an IgG, a PNA5 and a PBS, and a PNA5 and an IgG group. Now, why did we use this particular model? Well, rodents don't naturally develop Alzheimer's disease. However, these transgenic mice do express a neuronally derived human amyloid beta precursor protein, which results in them having amyloid beta plaque accumulation in the expected areas that we'd want them to see, such as the cortex, the hippocampus, the brainstem, and the cerebellum. So if we look in this picture over here on the left, all the brighter green would be the plaque accumulation on the thalamus. So how I went about answering this was I used amyloglow staining, and amyloglow is a fluorescent stain that binds to amyloid beta plaque. And then I used a confocal microscope to take images of the cortex and the hippocampus of the rat brain. And then I performed an analysis on the number of plaque as well as the density of the plaque. And then my results showed that there was no difference between the PNA5 and the vehicle. So between this black group and then within this green group, and same thing on this graph as well, that there was no difference. However, there was a difference between the PBS and the anti-amyloid beta antibody treatments. So between the black group and then the green group, there was a difference, where the anti-amyloid beta antibody had a higher plaque count and density, which is shown in these representative images above, where the PBS, both of the PBS situations actually have less of a signal, as opposed to the anti-amyloid beta antibody IgG situations, which both have a higher transmitted signal. Now the next steps that I plan to take is that I want to further read about ARIAs, and I want to read some prior research to see and compare to my own conclusions and results, and I want to complete a data analysis of an entire animal brain and an entire hippocampus. And the remaining questions I have are, does the immunotherapy reduce amyloid beta plaque? Does PNA5 improve the immunotherapy? And does PNA5 affect amyloid plaque content? And these are the same questions I started out with because I want to try and rerun my experiment using a higher N number. And I also plan on doing a full analysis of the hippocampus and the cortex of the brain to find out if my results were confirmed. Some other fringe benefits that I got from this SIMI program was I got to meet some wonderful people, I got to participate in lab meetings and journal clubs, and I learned how to use lab machines, tools, protocols, and etiquette. And last of all, I'd like to thank all the wonderful people in the PyRes lab that are over there, and then the wonderful people that are in the SIMI program for allowing me to take part in this summer. Any questions?
Anything coming in the back of the room. So if you want to make a comment, mention, would you come up to the front to just comment? If you have any additional comments you want to make. Because we're not picking up the sound from the back of the room.
Sorry. I just speak loud.
Yeah, it doesn't work. Loud isn't loud enough. It's okay, you're fine.
Well, it's good. Yeah. Sorry.
And speak loud.
Was a pleasure has Akash with us. Unfortunately, Dr. Quinn not able to come today because he had another meeting. But Akash is a really good student. He always come with brilliant question and work really hard. Those days part from a project that we try to create a model to study the mechanism of ARIA in Alzheimer's disease. And Akash know that he had a lot of image to do analysis, so he was able to perform part of the analysis. And he did a brilliant work, and it's wonderful having him.
Yeah. All right, our next student is Rawan Albergash, and she is coming to us from Basis Tucson North, and she was mentored by Dr. Weinhoff.
Hello everyone, today I'm going to be talking about my summer in SIMI. So a quick overview about me. My name is Rawan Albergash. I'm 16 and I go to Basis Tucson North, and I'm a rising senior. In the future I hope to be either an OBGYN, a pediatrician, or a pediatric psychiatrist, as I really love children and I hope to work with them in the future. So quick project overview. My lab mentor is Dr. Weinkoff. Our lab's main question is, what is the impact of carotid disease on cognitive impairment on Alzheimer's disease? And this is important as it could provide us new ways for preventing dementia, as carotid disease can easily be treated through various lifestyle changes, surgical procedures, and different medications. So the questions I began with were, what is carotid disease? So when I was first introduced to the lab, I wasn't exactly sure what it was. But after doing research, I know now that it's the narrowing of the blood vessels that carry blood from the body to the brain due to plaque buildup. And so after learning about carotid disease, it led me to these next two questions. How does carotid disease affect cerebral blood flow? And how does that affect cognitive functions in the brain? So basically, my thought process was that if the blood vessels that carry blood from the body to the brain are narrow, then shouldn't there be less blood entering the brain? And if there is less blood entering the brain, then wouldn't that affect how the brain functions and how we think? So this led me to my overall research question, which is, will subjects with carotid disease have a lower score on the fluid cognition test than on the crystallized cognition test? So basically, quick overview of this is that we administer neurocognitive tests on all our patients, and some of those specifically test for fluid cognition, while others specifically test for crystallized cognition. And fluid intelligence is basically your ability to assess and evaluate new situations without any previous knowledge, while crystallized intelligence is accumulated knowledge over time that you can recall. So I was just curious about that, and if fluid intelligence would have a lower score than crystallized intelligence. My hypothesis was that subjects with carotid disease are going to have a lower score on the fluid cognition test than on the crystallized cognition test because research has shown that carotid disease has led to cognitive decline and Alzheimer's, and in many Alzheimer's patients, fluid intelligence is what decreases first. So that's why I thought that. So how I answer my question. I first split all the neurocognitive tests we perform on our patients into those which test for fluid intelligence versus those which test for crystallized intelligence. So what I did was I did research about each of those, and I found out that the definition for fluid intelligence is the ability to solve and do tasks without any previous information. And I also found out that it is primarily mediated by the dorsolateral prefrontal cortex. And then the definition for crystallized intelligence is accumulated knowledge you can retain. And I also found out that it's primarily meditated by the hippocampus and temporal lobe. So what I did was I did research on each of the different neurocognitive tests we give each of our patients, and I saw which category they fit into most. And I came up with this. So these are all the fluid intelligence tests and all the crystallized tests. And so what I did was I compared, for each test, I compared the scores that carotid patients would get on them and the scores that non-carotid patients would get on these tests. And I used this one statistical analysis called t-test to see if there was a significant difference between the two. Afterwards, I went back and I saw which of these tests with a significant difference were fluid and which ones were crystallized. So for the fluid intelligence, these are some of the tests that had a significant difference. So basically for t-test, it gives you a p-value after comparing the scores each patient gets. And if the p-value is less than 0.05, then you know that there is a significant difference. So as you can see here, all of these tests, the MoCA attention, trail making test A, and trail making test B, all had a significant difference. And these were also some tests for fluid intelligence that as well had a significant difference. And then these are some of the fluid tests with no significant difference. As you can see here, all the p-values are greater than 0.05. And then for crystallized intelligence, we had these two tests, which were had a significant difference between carotid patient scores and non-carotid patient scores. These are also some tests that had significant difference. And then these are some of the crystallized tests that had no significant difference. So again, all of them, all the p-values are greater than 0.05. So I was analyzing the results. I saw that 5 out of 9 of the fluid intelligence tests had a significant difference between carotid and non-carotid patient scores, while 4 out of 9 of the crystallized intelligence tests had a significant difference. And so this led me to question what is in common between the 9 total neurocognitive tests, so like the 5 fluid and the 4 crystallized. So what's in common between those? So analyzing our results, I found out that both fluid and crystallized are affected by carotid disease, and neither one is affected more or less by carotid disease. Like my hypothesis, I thought that fluid would be more affected than crystallized, but after analyzing our results, I found out that both are almost equally affected. And then after going through the tests that did have a significant difference, I realized that each of those tests required some sort of frontal lobe activation. So I concluded that carotid disease primarily affects frontal lobe function. So the questions that remain is why those tests? So why were those the only tests which were affected by carotid disease? Like how come the MoCA language test wasn't, or the category fluency vegetable test wasn't? And my second question was why was the category fluency animal test not significant while the category fluency vegetable test was? So we give our patients these two tests, and they're basically the same thing, but one is asking about animals and one's asking about vegetables. But after I did the t-test, I found that only the category fluency vegetable test was significant, and I was a bit confused as to why that would be, because you'd think that they're the same test, just one's with animals, one's with vegetables, there wouldn't be that much of a difference. My third question is that how come in Alzheimer's patients, fluid intelligence is more affected than crystallized intelligence is? And then my fourth question is just like a general overall question. How can I improve my lab, and what are things I could have done differently so in the future when I do more research, I can look back on this. So what am I going to do about these questions I have? So the first question, I want to do more research about each of the tests that did have a significant difference between the scores and find common similarities and dig deeper into them. And then to answer my second question about the discrepancy between the category fluency vegetable and the category fluency animal test, I want to read more into the different processes involved in memory and find out why some memories are stronger than others and different correlations memories have in our brain. And then to answer my third question, I want to research more into the exact ways Alzheimer's affects the brain and find how it correlates to each intelligence, so fluid and crystallized. And then to answer my fourth overall question, I want to look back on the summer and just really reflect on what I've done and what I could have done better, so in the future I know what I can change. So overall, some fringe benefits was that I learned how to properly read and analyze a research paper. When I first entered my lab, I didn't realize how hard it would actually be to, as a high school student, to read a research paper and understand everything in it, because they use very advanced terms that as high school students, like, we don't know this stuff. So now I know how to properly read them and what to look for and just like make the most of my time. Then I learned how to come up with valid, testable hypotheses. So my lab mentor, Dr. Weinkoff, had me come up with 5 hypotheses to present to him in order to come up with what my research question would be. And so that really helped me hone down my skills and realize what I want to do and how to make these hypotheses. And then another benefit was that I learned how to conduct statistical analyses and use various programs dealing with statistical analyses. So when I was analyzing the different scores for carotid patients and non-carotid patients, I had help from different lab members, and I got to learn about the t-test, and I got to learn how to use this one program called PRISM, which does the t-test for you and helps you out with that. So I know how to do that now. And yeah, thank you. Big thank you to Dr. Whitty and the SIMI crew and Dr. Weinkoff and my Weinkoff lab members.
Scott, you were in the lab. Do you have a question or a comment that you want to make loud so we can pick it up? So you can come up a little bit. Will you be
Just
presenting
an overall
next?
comment, I'd like to thank you for showing up each and every day. Like, she didn't miss a single day, showed up, didn't leave early, showed up for all the hours, all 9 hours every day. Ready to learn, just came open-minded and ready to learn, full of questions, and it was an honor to be able to play a small part in mentoring you.
Thank you. And you're studying to be a mentor, you said in your blogs. How to have your students surpass you, right?
Thank you.
How are we on time?
Actually, right. 2 minutes. All right, our next presenter is who we just actually heard from, Scott French from here at the University of Arizona, and of course he was also part of the Weinkoff lab.
Well, thank you for the introduction. So my name is Scott French, and I'm an undergraduate student with Dr. Craig Weinkoff's lab. And today I'd like to share my project entitled Endothelial Activation Contributes to Early Neurodegeneration in Extracranial Carotid Artery Disease. Oop. And I have nothing to disclose. Extracranial carotid artery disease is characterized by plaque buildup in the common carotid and internal carotid arteries just outside of the skull base. ECAD is a well-documented risk factor for the development of stroke and TIA and is treated clinically to prevent such events. However, increasing data suggests that asymptomatic ECAD, or ECAD without a recent history of stroke or TIA, is associated with cognitive decline and may increase overall dementia risk. One of the mechanisms contributing to this increased risk includes endothelial activation. Endothelial activation is an early event in atherosclerosis and is characterized by the increased expression of pro-inflammatory molecules on the endothelium. In the brain, this initial insult to the endothelium leads to blood-brain barrier breakdown and neuroinflammation, two mechanisms which act synergistically upon each other to increase risk for Alzheimer's disease and related dementias. Given that little bit of background, our study began with two main questions. First, is VCAM1, a biomarker of endothelial activation, elevated in patients with asymptomatic ECAD? And along the same lines, is VCAM1 associated with any early neurodegeneration observed in these patients? To answer these questions, we utilized baseline data from 80 participants in our ongoing longitudinal study to evaluate the contributions of asymptomatic ECAD to overall dementia risk. Each of these participants underwent a blood draw, allowing us to quantify their serum VCAM1 concentrations, as well as an MRI to quantify brain morphology, including surface areas, volumes, and thickness of various areas in the brain. To detect VCAM1 in the serum of our participants, we utilized commercially available sandwich ELISA kits. Sandwich ELISA involves adding a capture antibody into blank wells. The capture antibody acts like a glue that only sticks to VCAM1 but will leave everything else in the solution undisturbed. When adding the serum to the plate, the antigen binds directly to this capture antibody and becomes physically stuck to the bottom of the plate. After washing out the unbound particulate, we add a primary antibody which binds directly to the bound antigen, forming that antibody-antigen sandwich, hence the name sandwich ELISA. After another wash step, we add an enzyme conjugate which binds to that primary antibody and provides a binding site for a specific substrate. After our final wash step, we add that substrate to the solution, which results in a color change that is equal in proportion to the amount of antigen present in the solution. I've included an example of this color change here on the slide. We analyzed the color change using a spectrophotometer set to 450 nanometers, and we calculate the concentrations of VCAM1 using values obtained from a standard curve run with each experiment. For this particular experiment, the standard curve is located in the two columns on the leftmost side of the plate as shown here. So moving into the results, first we wanted to see if VCAM1 is elevated in patients with asymptomatic ECAD. To answer this question, we split our patients into two groups based off of their asymptomatic ECAD status. We find that patients with high-grade asymptomatic ECAD have significantly elevated VCAM1 levels compared to those with low-grade asymptomatic ECAD. Next, we wanted to see if VCAM1 was associated with any early neurodegeneration occurring in these patients. To do this, we compared VCAM1 to hippocampal volumes. We selected hippocampal volumes for this analysis because decreased hippocampal volume is one of the earliest manifestations of human cognitive decline and dementia. We find that increasing VCAM1 is significantly associated with decreasing hippocampal volumes. There are many potential contributors to decreasing hippocampal volume as well as VCAM1 release, so we found it pertinent to control for other potentially confounding risk factors in our analysis. After adjusting for age, sex, and various vascular comorbidities, VCAM1 remains highly associated with decreased hippocampal volumes. So in summary, we found that VCAM1 is elevated in the serum of patients with asymptomatic ECAD. Furthermore, elevated VCAM1 is associated with decreased hippocampal volumes, suggesting that endothelial activation secondary to ECAD may result in neurodegeneration that could be detectable in blood. Although the results are exciting, there is still a very important question that remains unanswered. Can serum VCAM1 be used clinically to stratify neurodegenerative risk in asymptomatic ECAD patients who are candidates for surgical intervention? This question poses clinical relevance as asymptomatic ECAD patients are currently only treated for stroke prevention, despite the increasing data showing that asymptomatic ECAD is associated with cognitive decline. A potential reason for this may be due to the lack of a clinically accessible biomarker able to stratify neurodegenerative risk in these patients. And so our current and future endeavors in lab are focused on addressing this issue by trying to answer this question above. Second, I'd like to thank Dr. Craig Weinkoff and the rest of the Weinkoff Lab for their support and hard work. I'd especially like to thank Sue Manzara for her help with data analysis for this presentation. And I'd also like to thank our sources of funding. And I'll gladly answer any questions you guys may have now.
Our next student is Lance Perez, undergraduate here at the University of Arizona and also part of the Weinkoff Lab.
Well, actually, I'm not part of the Weinkampf lab. It's Dr. Russell Whitty's lab. Okay. So Dr. Russell Whitty runs UNO, which stands for Experimental Ultrasound and Neural Imaging Laboratory. So our main focus is just trying to integrate ultrasound into novel biomedical imaging methods. So what is the issue my project tackles? Well, for a variety of neurological conditions like Parkinson's disease and seizures, one method of treatment is to insert deep brain stimulating electrodes into the brain. It's very important during this procedure that the electrodes be placed in the exact location that the neurologist has chosen, as to not stimulate a part of the brain that should not be stimulated at all. This can be done either during the procedure or after, using a variety of methods, most commonly computed tomography, CT scan. One of the problems with doing this intraoperatively or even after the fact of the surgery is that CT scans expose patients to ionizing radiation and are also bulky and interfere with surgeons being able to have a lot of room to work with. So a proposed solution for us to be able to map exactly where these electrodes would be in the skull during the procedure is to use something called ultrasound vibrational potential imaging. So a bit of a crash course on ultrasound vibrational potentials. When we place the metal electrode inside a tank of saline, which is just sodium chloride, there's a metal interface here, like a liquid metal surface interface, that there has to be a distribution of charges. Electrodes, there's a redistribution of the ions surrounding the electrode in order to neutralize the charge. This liquid surface interface is very sensitive as the ions reorient themselves, which leads to this static potential that decreases as we move away from the electrode. So when we pulse it with ultrasound, this obviously oscillates. We send an acoustic wave from the ultrasound to the electrode surface, which moves around these ions around the surface of the electrode, which creates a voltage that we can measure using the electrode itself. So my summer project, which was actually a continuation of my summer project from last year, I was in SIMI last year, was sort of last summer, I'll go into it, but it was locating electrodes through skull. So step one, we were trying, as a proof of concept, we wanted to locate the electrodes in just a simple setup, a tank of saline, so sodium chloride. So this is a summary of what I did last summer. We had our electrode array right here. Which was submerged in, like I said, a tank of saline, sodium chloride. We then pulsed the ultrasound to focus on specific sites, as you can see here, so we can get cross-sectional images of our electrodes. And we were actually able to pick up signals that represented these electrode sites. And with the known geometry, for example, each electrode site on this array was separated by 4 millimeters. So when we gathered our images together, we could compare it to the center-to-center spacing. And actually, we were able to get a 3.98 average center-to-center spacing between our measured voltages, which shows that we were sort of on the right track to knowing that we were both in the right location depth-wise, but also our measured voltages were also consistent with the separation of the electrode sites. So the second step in the project this summer was to see if we could locate these electrodes in a different environment. In this case, we had needle electrodes right here that were implanted in a skull cap, which was, I guess they were encased in an agarose and saline gel. So our main goal was to sort of have this set up where we would have our skull cap here with our needle electrodes implanted, we would couple the ultrasound transducer to the skull, and we would attempt to pulse the ultrasound through the skull, and like I mentioned before, oscillate the ion surrounding the electrode in order to induce a voltage that we can measure with the electrodes and be able to map the locations of each of these sites, whichever sites we were trying to locate in the skull, well, through the skull. So this is the bottom view. One of the things we had to consider was that the probe's face was sort of a rectangle, so you didn't want to orient it where you couldn't be able to, scan linearly across and get each electrode. So here's a visual of what it exactly looked like. Obviously we have the skull cap here. We actually have, you see this tape here, these are gold cup electrodes where we're trying to measure surface potential, which will become relevant later. But here we have the ultrasound probe coupled with an adapter in between the probe itself, the probe face, and the skull. So from there, the experimental procedure was to collect an acousto-electric signal to have a reference of where we would expect our ultrasound vibrational potentials to be. And then after that, we would perform a scan to collect these vibrational potentials. So I know I just mentioned acousto-electric signal, which was relevant to the surface electrodes that were on the skull. So what exactly is the acousto-electric signal? Well, the acoustic electric signal is based on the acousto-electric effect, which just basically is when we have a current density, which could be, for example, a neuronal current in the brain, or when you have an electrode, a stimulating current. When you oscillate the material surrounding this current with ultrasound, the pulse of ultrasound, the interaction of it with the material surrounding this current in the brain, creates a measurable voltage. So from there, our first step was to perform a scan where we had two dipoles, which was just like a positive and a negative, like at the ends of the battery, where current would flow from the positive pole to the negative pole. So we had two dipoles set up so we could get an image to sort of, like I said, get a reference of where we would expect these electrodes to be. So from there, we were able to collect AE signal, time varying as time went on. For example, the first dipole was this first needle electrode and the third needle electrode. And so from there we could see that we should expect to see a vibrational potential at around 3 in the axial dimension, which is relevant to like the face of the probe, and around a depth, which was from the probe to about where the skull was, of around 48.2. And as you can see here, it's just further AE scans showing the two dipoles now at the same time in reference in time. And then we could also see that our second dipole, well actually, scratch that, it wouldn't actually be a so this is just showing two electrodes. It's not exactly showing, for example, here we have 1 and 3, our positive and our negative, but it's just showing one dipole. So a further question I would have was sort of figure out a way we could figure out which one was it exactly. So from there, after we got our AE data, we were able to perform an ultrasound vibrational scan. So at this point, each of these needle electrodes were just recording for voltage. They were not stimulated at all. No current was leaving from them. And we were focused on the fourth one right here. I know it's kind of hard to see, but yeah. And so from there, actually, an interesting thing is that you would expect to see, so if we were focused on site 4, we would expect to see a signal around the depth that we had between the probe and the electrode array. We would expect to see like 4 here, 3 here, 2 here. We were actually only able to pick up on all of them a bright signal at around negative 12.3 in the azimuth direction and 44.7. So a further question I would have for this is sort of discerning why we were only able to, on all 4 electrodes, were we able to pick up the same signal, and we weren't actually able to locate exactly where these electrodes are. We weren't able to collect ultrasound vibrational potentials, or maybe not of a strong magnitude, with the electrodes themselves. So for further applications, I just wanted to show a quick little in vivo pig study of where this was actually done in person, where a DBS electrode was implanted into the hippocampus of a pig subject. And there was these DBS electrodes you could either stimulate meaning emit current from them or record from them. This is actually the exact same probe we were able to use for the experiments previously, which is just a 0.6 megahertz 2-dimensional ultrasound array, which was coupled to the skull of the pig. There's the gold cup electrodes, which are used to measure the acousto-electric signal that I mentioned before. And from there, It was good to see that there was, we were able to pick up these ultrasound vibrational potentials through the pig skull, which shows that perhaps the application of this methodology in humans is viable and could lead to further applications. So now conclusions and future work. So future work and further questions. I mentioned a few further questions I had before, but a major question I had was more so to do with like the signal processing. So it was nice to see that we were able to get these nice images, but I would really like to go back and understand exactly what exactly in the MATLAB code, what was implemented in order to not only just figure out where the voltage was depth-wise, but just the magnitude and all that and other metrics. So yeah, that was. Thank you for your time, and if there's any questions, please ask them.
Consider the questions for, there's several other presentations on the video. Thank you, Lance. And Lance presented earlier work at an NIH conference in Orange.
All right, our next student is Emilio Armenta. He's from Yuma Catholic High School, and he is part of the Russell Whitty Lab as well.
Hello, my name is Emilio Armenta. I'm going into my junior year of high school at Yuma Catholic, and this is my presentation on melanin mimicking tissue phantoms for flow acoustic imaging. This is a general overview of what I'm going to talk about in this presentation. What my lab, the name of the lab I'm working in is UNO, and what the lab is about, what my project is about, and questions that came along with it. The lab that I'm working in that I was working in for the summer is called UNO, and UNO is a hybrid ultrasound lab. So UNO stands for Experimental Ultrasound and Neural Imaging, and in UNO we have two types of imaging mainly. Well, basically just two types of imaging. That being photoacoustic imaging and acousto-electric imaging. The overall goal that UNO's photoacoustic team kind of has is for early melanoma detection, while the current goal that we have at least is to mobilize our photoacoustic imaging system and collaborate with Dr. Clara Curiel, who is a dermatologist that we're working with. And the goal of my project is to make fake skin that mimics melanin. The issue of melanoma. Melanoma is the deadliest type of skin cancer. It has a fast growth rate. It's very common and frequent, especially in regions like ours in the Southwest and in states like Arizona. The first questions I had when I found out my project would be about mimicking skin cancer and melanin were how can we detect skin cancers, what materials best mimic melanin, and what is photoacoustic imaging. Photoacoustic imaging is an imaging modality that uses a laser to heat up the skin, and when the skin heats up, it expands, and that creates waves that an ultrasound transducer can detect. This is just a photo example. This isn't what our photoacoustic system looks like. These are some things that photoacoustic imaging can do. It can detect blood oxygen levels. It can image in real time, and it makes an image based off how much light is absorbed. This is our probe that we use. This is a motor that lets us move the probe up and down. There's magnets at the end of this, so we can connect it to different things. Like when we're imaging on people's arms, we use magnets and we use washers, and we connect it to their arms so that, well, we can connect it to this magnetically. This is the